Good morning. Today we have planned the presentation in this way. First I'll be covering the facilities and regulatory compliance of CCM and Bsl-3 facility, then resources and services, how to avail the Center for Comparative Medicine Services, and then talk a bit about the animal model, and then Anupama will talk on the rest of the topic like research support, animal models, academics training, service charges of CCM, and acknowledgement. So let's first start with the journey of Animal House Facility to become a Center for Comparative Medicine with the vision of Professor Sareen sir and continuous guidance from him. So we got registered with the CPCSCA, that's a government statutory body, in 2011, and at that point we were registered for research for education purpose. And you can see in 2012-13 we have started operation in a small in upper basement of a ILBS in a phase 1. And at that point, the facility looks like this. And we were having a 60 rats and 150 mice capacity. Let's say it is X. And then eventually we have maintained that for almost 5 years. And 2018, by then, we have the phase 2 ready. And again, it's a proud moment. We are having a really very good double corridor clean and non-clean kind of huge ground floor facility. And that has been made functional in 2018. We have put the efforts, and you can see by 2019, our statutory certification or licensing has been increased from research from only education to we can do research for commercial purpose, we can do breeding for in-house, we can do breeding for trade, and this S represents for the small animals. So in 2019, we have started the in-house breeding program. We got our first lot of kind of IVCs, individually ventilated cages, and capacity expanded to 2x, and you see how the facility looks like in 2019. Then 2020, we have seen the Dr. Dinesh and Dr. Savneet's lab. They have increased the usage of rat, and then we have envisaged in 2019, okay, we'll be in need of. So we have just parallelly started to get one free of cost internationally first time rack in animal facility, and this is a rack which has increased our capacity to 3x for a rat. And then in 2021 we have added so many mouse strain, like Dr. Anupama has requested a GFP mouse, which is a kind of a we need to get an MTA from Jackson lab, and we got it from NII, and we have bred them. Then C3H for Dr. Ashok Kumar's lab, and we have done the DAN model there. Then B6N mouse, this is the black on a background C57 black 6N mouse, and long Evans red. These two strains we got it recently and expanding. So in 2021 we got another lots of cages, and we have expanded our capacity to 4x. So sorry. And then at the same time we have started the new model system Zebrafish in a static tank to just start with, okay, how the science is developing so fast, and different animal model systems are used, and especially Zebrafish is catching up next to the mouse. So we have under the guidance of sir, we have started this facility, and we are seeing that in 2022 we are going to add these 3 more things into the CCM portfolio, that is immunodeficient mouse experimentation, in vivo imaging, and we would like to move on for a zebrafish experimentation. So now this is again this is a very regulatory environment. So as a quality statement at ILBS, we believe that good animal care and good science go hand in hand. We achieve our belief by good welfare, human treatment of animals in our care, implementation of basic principles of 3 R's, which 3 R's means first is replacement. Can we replace the animal? Use. Second is reduction. Can we reduce the use in a research? And third is a kind of refinement. Can we refine the processes so the pain can be reduced? And these things can be done through IAC and veterinarians. Compliance with national rules and international guidelines. We have the national guidelines and we aspire to follow the international guideline as well. Now the backbone regulatory framework is this. This is come under the Ministry of Fisheries, Animal Husbandry and Dairy Science, the dairying, and CPCSA falls under it, and Institutional Animal Ethics Committee under the guidance of a chairman, Professor Ganguly sir. This committee regulates the ethical part of the animals, and it is as comparable to the Human Ethics Committee. At times, if we consider the larger animal, it becomes more difficult to conduct a study and get the approval of ethics committee, animal ethics committee for large animal studies. So how we, as IAC, we accomplish this regulatory compliance? We have IAC regular meetings to approve the protocols, then annual inspections performed by, and we have almost 34 SOPs driven animal care and use program. And IAC ensures that all the investigators, those who are working, they are properly trained and they are competent to perform to reduce the suffering of animals and get the good result out of it. Currently, we have 44 protocols approved for the IAC. Now, we have started from a small animal house facility and we are aspired to become animal care and use program. So basically, animal care and use program has three facets, like attending veterinarians and the staff, then investigator, then animal ethics committee, and supported by administrator. If each component must make its contribution so that the whole is greater than the sum of the parts. This is what our ambitions. And here I would like to say the ALAC International is a non-profit organization which accreditates this kind of programs internationally, and that gives a kind of a value to the science and research, and where science and responsible animal care connects. In India, at this point, around 29 institutes accredited, and this has a cost involved, and most of the institutes, they are commercial, around 27 institutes. So now switching the gear to Bsl-3 facility. So what infrastructure we have? We have 900 square feet facility in a phase 2 on the same ground floor. Behind our auditorium, we have the facility. This facility has been commissioned and equipments and handover happened in May 2019. Then we have started operations. Coincidentally, pandemic comes and we were fortunately with the vision of a search, we were ready with the Bsl-3 and we could use it immediately. In February 2020, it has been used for SARS-CoV-2 research by virology department. And this facility is continuous under the comprehensive annual operation and maintenance contract for 5 years. It is a cost-intensive operations, and these are the responsible officers, scientists in charge, Dr. Gayatri ma'am, Dr. Nirupama ma'am, Dr. Ekta ma'am, Dr. Pratibha Kale ma'am, and biosafety officer, Dr. Chagan, and I have been given a designated responsibility for Bsl-3. So we put all together, and this facility become very much useful for a community and during the pandemic hours. So why Bsl-3 needed? Especially risk group 3 agents, high individual risk and low community risk kind of agents, and those who have aerosol potential to spread the disease, they are those kind of agents required to be handled in Bsl-3 facility. At times, Bsl-2 agents also required to be handled when there is a high volume and some other procedures where aerosol can be generated. So now, just to show you the facility, ILBS has put in the, I would like to say that this is the one of the best facility in India, Bsl-3, in that we have restricted entry, and you can see here we can see the temperature, humidity, air pressure, everything is controlled, monitored with the CCTV, and negative pressures, minus 45 pascals, these are the basic equipment. And safety point of view, we have the air showers, emergency air showers, and everything should come out after autoclaving, so autoclaving provision is there. And the bottom line, and very important, is building management system. We can see everything from the outside, control from outside, any emergencies happen can be handled from outside, and pressures can be controlled, so nothing can come out. That's why it's a containment facility. So now, One is the engineering standard. ILBS has given us a wonderful engineering standards. Now ownership is with us to put the processes in place to follow the same thing, because safety is very important for own sake as well as community, and in by and large we can say for institute and the nation also. If breach happens, it gives a bad name, so we have to be careful. So we have put in place the SOPs. Then for getting access, we have the formality, we have to put the fill up the forms and the manual. These are the mandatory things users have to take first, and then only the access is given to the facility. Now again, the regulatory framework is more stringent than the animal one, because here the biosafety is involved. So this falls under the Ministry of Science and Technology, DBT, and RCGM, the Review Committee on Genetic Manipulations. This committee is a central committee like CPCSA, and then institute level we have Institutional Biosafety Committee. So now with my understanding, I would like to draw that attention that IBSC has the recommending authority for the Bsl-3 related agents experimentation. They recommend to RCGM, and finally RCGM gives the approval, and then we can conduct the experiment. So this facility has been built with the expertise from external experts. Then IBSC Bsl-3 lab certification, we have already submitted our application. We got couple of time more information requested, we have submitted and expecting. Prepared the SOPs and all, and recently we have initiated a formal training to comply with the regulatory requirements. So Bsl-3 may also be available for research use on a charge basis post-COVID time as per the institute decisions. It is very cost intensive, and we are required to make it more revenue generating as well. So now coming to my next thing is resources and services what we are offering in CCM. So on intranet we have shown this. Okay, we can provide as a CCM animals first. We can provide the animals. We can provide the resources. We can provide the veterinary consultation, and if Bsl-3 comes in, we can provide the animal biosafety level facilities for mice research. And we are committed to provide the services eventually when we have more things to the external customer. So currently we have in-house breeding program where you can see we have this many rat and mouse strain. We have started with Spross Dolly rat and C57 black 6 mice. Now you can see we have a 4 mouse strain and 2 rat strain. And it user need to keep in mind that when they have to plan an experiment, it takes, because of the biological thing, it takes almost 3 months to get 6 to 8 weeks of animals. It takes three months planning. So please, you are requested to plan the things advance. And we have the Zebrafish as well. So now step 2, avail the CCM services. Very simple 5-step. You have to plan your animal study, then prepare, submit, and get the approval of IAEC protocol from Institutional Animal Ethics Committee. Raise the advanced animal indent for the approved animal protocol. That, as I explained, it takes time, so you have to plan well in advance. Schedule and conduct an animal study with the veterinarian in the facility, and finally the billing and payment. So this will be covered more by Anupama in the next session. These are the animal models we are having in CCM. Most of the models will be covered by Anupama, and I'll be covering the Zebrafish one. Zebrafish is a kind of a new model system coming in, and it is kind of just to appreciate the size. Their embryos are 0.7 mm and the larvae is 3 to 10 mm, and adult zebrafish is 2.5 to 5 centimeter. So now this was one of the Nature publications where, because of these reasons, like they have 70% protein coding, human genomes are similar in have the ortholog in zebrafish, around 80% gene associated with human diseases have the zebrafish counterpart, and more than 40,000 publications have come up, and major research area listed here, in that the zebrafish has used. So if you see in this Venn diagram, there are 73 genes which are exclusively common for human and zebrafish. They can only be studied in zebrafish. There are 2,000 around genes, they can be studied in zebrafish, mouse as well as human. So there's a lot of similarity, a lot of potential is there. Here the advantages are listed. This zebrafish model comes in between the in vitro system where we are studying the things in the cells, and then we are going into the mouse. But in between, to get more understanding with the high throughput, the system is very much helpful, and they are the vertebrate animal. They have the kind of ease of husbandry, less expensive. Their embryos are very transparent, and through imaging, even we can track a single cell, where it is going, what it is doing. So molecular mechanism understanding is also very amenable in this model because of the optical clarity and imaging development. Then molecular conservations of development, and there are some disadvantages. Every animal model has some disadvantage, and scientists are working to address this disadvantage eventually, but some of the anatomical and physiological disadvantages may not be addressed. So that is the limitation of this model. So now, to get the understanding of it's a biology life cycle, from embryo to within 72 hours they hatch, and most of the research, like the high pace research, in this stage embryos are very transparent, and we can see all the cell movement, study the molecular mechanism. That's why most of the research is done in a larval level stage, and especially for our liver related research, where we expect the chronicity. So still there are progress going on to study these things in animal research, in a zebrafish adult models. So liver is little different. They do not have a lobules and cord kind of structures, but though the zebrafish liver being a vertebrate, all the cells available, they are scattered and almost functioning in the same way the human liver functions. So it gives the important information. So now just give you we have the confocal imaging facility. So how beautifully, this slide I would like to show that how beautifully even single cell, different cell we can track in the zebrafish. First, this is showing hepatocytes. The line itself is reported for hepatocytes. Then intrahepatic biliary cells, you can see here only those cells. You can study those cells. And third is endothelial cells and hepatic stellate cells. So all individual single cell type also can be studied. So now what we have put it at Zebrafish facility at ILBS. So we have set up the things. Primary, we are able to maintain adult and larval fish, and we have done the histopathology setup, basic processes. They are little different from the mammals, and then we have set up those things with Dr. Chagan. Oral dosing we can do. There are 3 PIs interested, Dr. Savni, Dr. Gayatri, and Dr. Nirupama, and like we have submitted one grant with the Dr. Savneet, and in the collaboration with Dr. Reddy Institute of Life Sciences, still under review. And way forward, we are saying that okay, investigators should consider this model system. We are here to kind of collaborate with them and write the grant, get the fundings. We can plan experimentation and import and creating a new transgenic zebrafish line is the aspirations. Now this is how the facility is in, and we aspire to develop this kind of facility eventually. So now, I could not justify it more scientifically in 4 or 5 slides how the zebrafish is useful. So these are the 2 review articles. One is recently published in 2021 by very reputed Institute 3, and in that they have reviewed why the zebrafish is useful, and specifically for liver also they have listed okay, for liver how the zebrafish model can be used. With this, I'm passing the presentation to Anupama.
So, before coming to my topic, research support and animal model, I would like you to emphasize that there are factors which governs the biology of the animals other than the genetic factor, most importantly environmental factor, which PI may not appreciate or see, but this could lead to erroneous conclusion in their research. So to control this environmental factor, CCM maintains strict standards for macro as well as micro environment. So macro environment is like housing condition of animals, like room temperature, relative humidity, room air changes, noise, etc. And micro environment, which is surrounding of the animals within the cage, like housing condition, that is individually ventilated cage system in which we are doing the housing of the animals. And bedding, we use autoclaved corn-cob bedding, and then we give standard feed which is gamma irradiated, and we give autoclave water only. So CCM maintains all these standards so that your in vivo research is more reproducible. CCM staff has expertise in all this to support the in vivo research, such as blood collection techniques like retroorbital sinus blood collection, saphenous vein collection, then cardiac puncture. We have also expertise in dosing by various routes like intraperitoneal injection, intravenous injection, intramuscular injection, and also drug administration by oral gavas and intranasal. We have also expertise in necropsy techniques for collection of various tissues and organs, such as liver, lungs, pancreas, brain, etc. We have also expertise in surgical techniques like perfusion, bronchoalveolar lavage, peritoneal lavage, then ligation of common bile duct and cecum to generate sepsis model, then cannulation of arteries and veins, and then partial hepatectomy and engraftment of cell by intrahepatic or intrasplenic or directly intrafemoral. We also used to perform oral glucose tolerance test and insulin tolerance test for metabolic studies. So these are our expertise of animal care staff as well as of veterinarians to provide you support. Then these are the list of animal models which CCM has contributed and worked along with the various investigator, and these are the timelines to develop this model. So I will be discussing few models in my further slides. So these are the contribution of publication from the CCM. So last two publication is under review. So coming to the models, this is our first model which we have published by the Aditi Varsane in Hepatology 2018, where we have given HUH7 cell line, that is human hepatoma cell line, in nude mice, and where we have seen the tumor progression after 2 to 3 weeks. We have given some therapy in the nanoparticle cargo with the microRNA and 199-3P, and we have seen the tumor reduction. So to conduct these kind of study, it takes 6 to 7 weeks of time. So this xenograft subcutaneous mice model is well established in CCM. Then orthotropic liver cancer, we are recently working on it. In this model, we directly inject cells into the liver, and tumor progress within 3 weeks after surgery, and here you can see the histology of tumor and non-tumor region. So the both subcutaneous and orthotropic xenograft model is well established in the CCM to give you support. Then coming to the next model, ethanol induced model, which we have worked with Sudhishti by Dr. Shwetang lab, and in this we have given a liquid diet to the mice rather than the regular chow diet. So it is required to be acclimatize the animal on the liquid diet. So this liquid diet contains ethanol up to 25%, and after giving this diet, animal were seen to up to the 12 weeks, and liver disease progression was seen. So after the progression of disease, we have studied histopathology, liver proteomics, serum biochemistry, histopathology. So and we have also checked the histology and fibrosis score by MT in both the control and ethanol group. So here you can see the biochemistry profile of the ethanol versus control, where the fibrosis score is higher in this ethanol group, and LT is higher in the ethanol group versus the control group. So moving to the next slide. In this, we have developed hepatopulmonary syndrome model, and we have given fingolimod intervention. So we have developed this HPS model by CBDL, that is common bile duct ligation. So after 2 to 3 weeks, there is a progression of HPS, and where we have given the intervention and we have observed the animal further 2 weeks. So here you can see the development of hypoxemia, where partial oxygen level is low in CBDL group versus non-treated and sham group. More albumin influx we have seen by measuring the Evans blue vascular permeability assay, which is higher in CBDL group. Then we have checked the lung versus dry weight ratio to see the pulmonary edema, and we have also checked the nitric oxide level in bronchoalveolar lavage. So this is higher, you can see, in the CBDL group, that is HPS model, than the sham and treated. So we have also seen the liver injury to validate our model. So here the ductal reaction and inflammation you can see clearly there in the CBDL HPS group. The survivability of normal CBDL HPS is less than 50% up to the 30 days, but there is more survivability when we have given the fingolimod intervention in this HPS model. So it is up to around 100%. So this is under a review. So moving on to next, which we used to do is 2/3 partial hepatectomy. In this we are studying the bioenergy, metabolism and role in liver regeneration along with Dr. Anupam lab. So here we ligate the 3 lobes, that is left lateral and median right and left lobe, and we dissect these lobes, and here the mice is after the resection. So this liver mass reconstitute within 7 days, and so the model developed within 1 week of time. So this kind of study can also be done. Then moving on to the next model is cecal ligation puncture model, where the severity of sepsis determined by the length of the cecum ligated. So when we ligate more than 2/3 of the cecum, then there is more severe sepsis. So here you can see after the ligation we puncture the cecum with the 20 gauge of needle, and there is cecum necrosis and mortality after in the 72 hours of time. So the timeline to study this kind of sepsis model is around 72 hours. So these are some of the ongoing CCM projects on animal model which we are performing, like HCC model by Den induced HCC, in which we are studying mebendazole and lenvatinib drug combination therapy for HCC, then bacterial exosome therapy for a xenograft model to target specifically the HCC cells. Then we are also studying exosome and cell therapy in advanced liver disease like HE model and hyperammonia model, and also in advanced liver disease model. We are also performing pharmacokinetics and acute toxicity study like IR415, which is an anti-HBV compound, and we are also performing autoimmune hepatitis model by bacterial vesicles. So these are some of the model, and these steps already discussed by Dr. Patel. So here I would like to emphasize that when your protocol is approved, so it takes around 10 to 12 weeks of time. So this includes the mating time, gestation period, weaning, and maturity of the animal. So you have to plan your experiment accordingly well in advance, and you have to raise the advance intent. That form is available on the website also. So then after the study, that is according to your study duration, your billing will be done on monthly basis. And CCM also organize academic and training program for the PhD student as well as for the external students as well as faculty. So this PhD course, Laboratory Animal Methodology, Welfare and Ethics, we conduct twice a year for our internal PhD students, and we also conduct hands-on workshop training on basic animal experimental technique, and this is on chargeable basis. And we also give advanced animal experimental technique training if PI requests on chargeable basis. So we have so far trained 104 students, internal as well as external, as well as scientists in this training program and academic session. So these are photographs of our workshop conducted during 2020 and this in pandemic year only. So these are the charges. I would like here to say that there are two type of charges which CCM charging. That one is animal charges. So these are the cost for the animal, and these are the cost for the per diem maintenance, that is cage maintenance charges. So this charges is very, very subsidized for the ILBS user than the government institute user, other external users. And these rates are approved from the competent authority. Coming to the future prospect, so these are our short-term prospect, you can say. In vivo imaging facility, we are very near to achieve this. Equipment already arrived. We are in 1 or 2 months installation and training will be done, and this will be available to the faculty and users to the ILBS, and also on the charge basis for the external. Then immunodeficient, we have already validated the facility and we are waiting for some of the equipments, and breeding pairs we are going to order for this. So immunodeficient facility will be available. Then Zebrafish experimentation already told by the Dr. Patel. And in vivo contract, we have received few research project, but we are looking into it more, that is more executable at our facility, because we have received one in which behavioral study is required. So that facility is not available with us. So we are looking into which is more executable with our facility. So, and these two, which is our long-term prospect, and we require funding and project in this area to develop. Then coming to the end of my presentation, I would like to say that good animal care and good science go hand in hand, and investigators are encouraged to use the CCM service to answer their clinical translational basic research question which may need prior in vivo understanding, and veterinarian and animal care staff are committed to support the research endeavor in their best possible way. So I would like to show you the CCM team. She's a scientist in charge, Dr. Savneet, and these four are our efficient, dedicated technical staff, Karamjeet, Krishan, Abhay, and Mr. Satyendra, who has joined recently. And these are our collaborator and user of our CCM. Then I would like to acknowledge Sareen sir for their guidance, and Dr. Gangli sir for their guidance in IAC as a chairperson. I would like to acknowledge Dr. Nirupama and Dr. Gayatri, Dr. Savneet, and from Dr. Virology, from Dr. Ekta, external experts, member of our ILBS committee, IBSC, IEC, and CCM service, and our engineering and medical operations staff to support our facility daily operation. Thank you so much. So I would like you to take a virtual tour to experience our facility, as you might not have opportunity to see the our facility. So this is a restricted area. So I'd like you. So this is our phase 1 facility. This is one room, animal holding room and procedure room was there in phase 1, from where we have started. And this is our new phase 2 facility. So here you can see the traffic and how we do the PPE. This is our clean supply area. All the material comes after autoclave, and this goes to the clean material storeroom. And these are our clean corridor, and one side is the animal holding room, other side is the procedure room, 4 procedure room. This is our animal experimental room in the individually ventilated cage system. And after use, this cage goes to the dirty corridor. This is our animal procedure room. All the procedure done under the biosafety cabinet only. So this is our rat breeding room. Then this is zebrafish room, static tank, adult fish, nursery, then atremia culture, zebrafish mating on marbles. And this is how we exit from the facility. So these are our support areas, feed and bedding stores, and washing area. We have double door autoclave. So these are by-waste area. This is dirty corridor from where Bsl-3 and all the animal facility by-waste comes. So that's it. Thank you.